Hi everyone, I'm Kiki and joining me today is Matt. We're both graduate students from the Department of Molecular Genetics at the University of Toronto. This year, the theme of Science Rendezvous is STEAM Green, so we thought it'd be a great idea to combine genetics with sustainability. That's why we want to talk to you about GMOs. Have you heard of a genetically modified organism, or GMO? What do you think it is? Do you think a GMO is A, something scientists use to study the world, B, all living things, C, living things with its trait changed by humans, D, living things with its trait modified by humans in a lab, E, all of the above, or F, none of the above? If you pick C, living things with its trait changed by humans, then you are correct. If you're wondering why C is the answer, continue watching this video to find out.
Hey everyone, I'm Matthew. So let's get into things. Now GMOs are defined as using genetic engineering or really any other process where traits you want are introduced into a specific organism. And this can be done through two methods which we'll soon go over, which is selective breeding and directly modifying the DNA of the organism. Now in some parts of the world, like in Europe, organisms made by selective breeding actually don't count as GMOs, but it's really different depending where you are. But here in Canada, a GMO can be made by both methods. Now let's quickly go over the oldest method of making GMOs, selective breeding. To quickly explain, all living plants and animals will have individuals that are just a little bit different than each other, and that's based on pure chance. Whether that is some are a little taller than others, some are a little more yellow, or others just need less water to grow. What humans learn to do is to pick the individuals that are different in a way that is desirable, like having slightly bigger fruits. They allow then only these individual plants to reproduce and make many new ones. And from these new plants, they once again pick the individuals with the biggest fruits, and this process repeats again and again, and over hundreds to thousands of years, the plants and animals we use for food today came to exist. Now let's check out some plants that we eat today and see what they used to look like. Alright, so here I will be showing you what a plant used to look like, and you will guess what the modern day version is. Here's our first plant on the left. Take a few seconds to look at this carefully and think about what this may have been bred to become. And it's a banana. Did you get it right? Well, on the left is the original wild banana, which was what it was before humans bred them to be far more delicious to us. So for our second plant, what do you think this may be? It's corn, and it's gotten a lot more appetizing over the years. So now, what may we have here? If you guessed this one, really good job. It's a carrot. Hard to tell without the orange for sure. For our final plant, take a crack at guessing this one. Surprised? No, they are not oranges or peaches, they are actually pineapples. Quite the change there. Now, let's move on to more modern methods of making GMOs. Directly modifying the DNA of organisms in the lab lets us make GMOs extremely fast, far faster than selective breeding ever could let us do. The first technique developed, and most common still, is adding DNA from other organisms into the one you want to modify. There are essentially endless possibilities with what you can put in here, including making bacteria produce insulin for treating diabetes, making plants repel insects so you don't need to use as much pesticide, and even making fruit take longer to expire or go brown. Some even more recently developed techniques that have been really improved on in the last decade are removing or changing the letters of the host organism's DNA. These can change the functions of genes to lead to a specific trait to appear, and doesn't require any DNA to be used from a different organism. It's all the original host. So it may seem pretty awesome that we can just change what characteristics plants and animals have. But why would we want to make GMOs besides it being cool? There are so many reasons, but some of the biggest are being able to feed more people with better food, reducing the levels of waste and greenhouse gases made, which is excellent for the environment. GMOs are essential for scientists to perform their research. GMOs can also make better medicine easier to make. And finally, we can use GMOs to help prevent the spread of disease.
Now that you know what a GMO is and why they're made, guess what is the world's first lab-made GMO? It was actually a bacteria that every modern scientist currently uses in the lab. This bacteria is called E. coli. In the 1970s, Dr. Stanley Cohen and Dr. Herbert Boyer were the first to show that bacteria can talk by exchanging DNA through something called plasmids. During their many experiments, Dr. Cohen and Dr. Boyer created the first kanamycin-resistant bacteria. This means that the bacteria has been engineered to grow even in the presence of a drug called kanamycin. Their work was not only a breakthrough in science, but it also kick-started the entire biotechnology sector. Since the 1970s, food scientists have made further progress. That's why I would like to tell you 2 stories about present-day lab-modified GMOs, and Matt will tell you about 2 future GMOs. So here's the story of how a GMO papaya called rainbow papaya saved the Hawaiian papaya farming industry. In the 1990s, a virus that infected papayas called the ring spot virus spread to almost every farm in Hawaii. By 2000, papaya production was down by 50%, and the industry was in great danger. Then, Professor Dennis Gonçalves and his team, who was working at Cornell University at the time, created the rainbow papaya, which was engineered to express a protein that prevented the ring spot virus from infecting papayas. After intense scrutiny by the USA's government, it was approved, and farmers began planting papayas in 1998. By 2015, more than 70% of papayas imported from Hawaii were rainbow papayas. If you are interested in how Professor Gonçalves and his lab made the rainbow papaya, you can hear it from the man himself in this video linked here. Do you notice how when you cut an apple and leave it for some time, it begins to turn brown? Many people find brown apples to be unappealing to eat and thus decide to throw them away. This generates a lot of waste. That's why a Canadian firm called Okanagan Specialty Fruits genetically engineered apples that won't turn brown. They called them Arctic apples. They did this by introducing a gene that would act like a light switch. This gene could turn an apple gene called PPO into off mode. In Canada, these apples are only sold in slices. However, out of all GMOs, these Arctic apples remain highly controversial. If you're interested in why that is, I encourage you to research it yourself.
Those are definitely some interesting GMOs that Kiki just showed you. Now let's go over 2 GMOs you may be seeing in the near future. So how many of you have heard of the Cavendish banana? Well, if you haven't, you've most likely tasted it before, since it is the banana we've been eating for decades and is the main banana sold everywhere around the world. It wasn't always this way though. Before the 1950s, the Gros Michel banana was the main banana sold. Your parents or grandparents may remember it. The Gros Michel was actually sweeter and had a thicker skin than the Cavendish. The Gros Michel, unfortunately, was almost completely wiped out in the 1950s because of Panama disease, which is caused by a fungus that spread to all major plantations. The Cavendish, however, was not affected by Panama disease, and farmers quickly switched to it. But today, a new fungus called TR4 has appeared and is very good at destroying Cavendish banana trees. It's quickly spreading around the world, and if nothing is done soon, we will need a new banana species to farm. Thankfully, scientists have been able to make a GMO Cavendish banana that can survive TR4. Nematodes, as well as a banana species called Musa acuminata, have a gene that can be added to the Cavendish DNA and make them resistant to TR4. In the picture here, you can see how unmodified Cavendish trees die, and bananas go rotten when infected with TR4, while the GMOs are totally fine. Hopefully these GMO bananas are soon shown to be healthy to eat for us and can be grown to keep our bananas the way they are now. For our second upcoming GMO, it's important for you to know that when you're told to eat your vegetables or whatever food because they're healthy for you, there's a good reason for it. Humans get a lot of the important vitamins and other nutrients from these specific foods. Sadly, many people around the world, especially the poor, cannot get enough of these to stay healthy, and this is a big problem. Scientists have turned to the yeast Saccharomyces boulardii to try helping here, which can be seen on the bottom picture under a microscope. That's what this yeast looks like. This is the only yeast species that can survive inside of our digestive systems. Because of this, it's used commonly as a medicine actually to prevent diarrhea and several other gut infections caused by bacteria. This yeast was modified by inserting 4 genes into its DNA that together would allow it to make beta-carotene, the molecule your body directly turns into vitamin A, and is the same molecule found in carrots, which is also the same molecule that makes carrots orange. They were then able to take this GMO yeast and put it into the intestines of mice. There they showed lots of the molecule was made, which really was a great success. Their next steps now are to show that the mice can absorb and use it to make vitamin A. It's a great demonstration of what can be achieved with GMOs, and perhaps this yeast will be given as a medicine to treat people suffering from the lack of a specific vitamin or other nutrient.
Now that you heard about present day and future GMOs and why they were made, let's talk about sustainability. What do you think is sustainability? Is it A, maintaining the balance of our ecosystem? B, meeting our needs without blocking the future generations' ability to meet their own needs? Or is it C, all of the above? The correct answer is all of the above. Considering what we have talked about today in this video, do you think GMOs can help in maintaining sustainability? Take some time and perhaps talk with others about this. Before we go, Matt and I would like to give you a challenging but also fun question. If you were a food scientist, what kind of GMOs would you make and why?